11 kilobase cluster. It's very small. At that point, I had a research project, Eric Schmidt had a research project in the States, and we were fighting each other for 5 or 6 years. And we published nothing for 5 years. But then I was working with my colleague, Jim Naismith at St Andrews, who's a crystallographer, and we did the first expression of the single proteins and the crystal structure. So that wasn't this one, it was the second one I'll show you, but the point is I'm going to explain it in a logical sequence. But in 2012 I was able to present simultaneously the 2 genes and the crystal structures that we had produced at that point in time, which put us a long way in advance because they were the first crystal structures of this type of enzymes to be published at all. And you can see why it's so important because of what we can do with it. So the heterocyclization enzyme takes this very simple sequence and then it puts in either 2 heterocycles where the cysteines were, or it puts in 4 heterocycles where the threonines and the cysteines and the serines were. The only comparable enzyme is Mitchell's system, which is the 2 enzymes that do the same process, but they're not very flexible. So what happens is these enzymes here only process when it's glycine, threonine, glycine, serine, or glycine, cysteine. In our case, we can process with large amino acids in front as well. So ours is much more flexible. It's a single enzyme as well. So we're able to get the crystal structure. And again, that's a major advance. It's the first crystal structure of this type ever published. And what we learned about it is that it works in a particular way. So you need some of the leader sequence to be able to process it all. And it can process very remotely. So a very strange enzyme. It holds here, right, very far away from the active site, and then it processes somewhere over here. So for biotechnology, that's a good sign. You can be very, very flexible enzyme because it can do things far away from the recognition site. So if you can decouple recognition from activity, you might have a very useful thing. So the enzyme itself, just the basic kinetics. First of all, it binds based on the very small sequence, the AYD. And it makes a single heterocycle. Once it makes that single heterocycle, it binds again, and it makes the further heterocycle. So it always goes from the C-terminus to the N-terminus. It goes backwards, as you might imagine. It's processive. It always goes that way, that way, that way, that way. It's quite interesting already to understand how this enzyme works. What it can do as well, it can accept non-natural substrates. So we added selenocysteine, and now we've got, selenozoline. It's hard to say. But just to prove that it can be done. So it was actually an experiment that was misunderstood by the postdoc, but he did it, and he got a great publication out of it. So it's kind of nice. I'm very happy he did the experiment. I wanted to do the selenium experiment because we couldn't find the product. And then he said, oh, I did it anyway, and now I can prove that I can make it. So actually, you can make unusual things with this enzyme. So this enzyme here is the least efficient and the least useful enzyme. What it does, it cleaves that there. So it cleaves at this end. It cleaves off the leader sequence. Now if you imagine, if that enzyme was really efficient, it would cleave off before the processing is taking place. So it needs to be really slow. So if you think about it logically, this enzyme needs to be bad. Because otherwise, it would cleave now, at this point, rather than at this point, when it's processed. So it's a very slow enzyme. We didn't know that at the time, but we did the crystal structure. So it's the first crystal structure we did. And this is a problem at the beginning, because Wael, who was the postdoc on the project, did the first thing, and you could see on the gel, you could see 2 bands all the time. The enzyme was processing itself, killing itself. That you can see in the crystal structure, it's breaking itself into pieces. We didn't know you could do a crystal structure on enzyme that was broken, but it still worked. But in the end, we made the inactive version of the enzyme. So we inactivated it by making a mutant, and then you can see the whole enzyme. And again, it's just a protease. Well, as I said, it's an inefficient protease. The catalytic triad is very bad. It's far apart, so it doesn't work very well. But what you can do with that information is you can look at how it might work. And again, it's just a protease. It just cleaves a peptide. But this is really interesting. So we had an organism called Cyanothef, which is which the gene cluster said it contains a lot of the same kind of gene cluster, a ribosomal peptide like the patelamides, called a cyanobactin gene cluster. And what you can see, it contains 10 substrate genes. So 10 precursor peptides. It should make 10 products. But it's finding those products that are really hard. So we looked at we modeled the substrate peptide, and then we modeled it in the active site. And we've discovered that this particular enzyme, parte, is slightly different. There's a little bit more space in there. There's more room. And it can cleave a different signal sequence. But now we have to try and find the product. And it didn't work. And again, it was accidental discovery. The room that we used for our cyanobacterial cultures is kept at 20 degrees and with light on. Somebody was going to do E. coli culture and switch it up to 37 degrees. And the heat shock killed the organism, but as it killed it, it produced the product. So something in the heat shock was able to make it produce these compounds. So, weird. And we were able to reproduce that. So when we did it, we saw this the first time. We just did a reproducible heat shock, take it up to 37 degrees, and then you see the product produced again. So something in the genome says switch on this production when the temperature goes up. And I don't know why. But again, these products were there. It was a real search and destroy mission. We had to find tiny amounts of molecule in a very big mixture and prove that it existed. So again, we got fragmentation on that. So I'm very proud of that very complex piece of research being successful. And the Americans didn't make it successful. Again, that's why I was happy. But anyway, let's go on the other enzymes. So this is the first big enzyme with biotechnological application. It's really cool. What it does, it takes the cleaved and processed substrate and it makes it into the macrocycle. So it does that step. And again, we were beaten by Eric Schmidt in the States. And he's a great scientist. I have nothing but good things to say about him. But he was always ahead of us. So that was the big problem. So what he discovered, he did a very bold experiment. What he said is that normally you have two substrates in the precursor peptide, which is why we find two in the original organism, again. And you can take one that's natural, VTAC, ITFC, and you can take one that's completely non-natural. And then if it's processed, you can check that it's worked, because this is already there, right? So you can change all the residues, as you can see. You can take away all the things that you think you might need, cysteine, serine, threonine. You can have the same sequence, AYD, AYD, and you get the same processing and cyclization. So this is the cool thing. You can make a cyclic peptide as non-natural. So he took an amphibian peptide, recoded it in here, and made it. So this thing, eptidendamide, is produced by the cyanobacterial enzyme in E. coli and makes a frog peptide. So it's really cool. But it can do lots of things. As long as the last amino acid is cyclic, and it can take D amino acids, it can take larger sequences, smaller sequences, etc. So this is really, really cool. And the mechanism is just an ordinary peptidase that's been hijacked. So instead of cleavage that you normally get, so for a protease here, it now keeps that stable for long enough for the N-terminus to come around and react and give you a cyclic product. So I think that's a very simple enzyme. And again, Eric proposed this. But so we were able to reconstitute the enzyme, so overexpress, reconstitute. So you can see now, you can get a cyclic product, you get 100%. This is a substrate that cyclizes very well. And just for people who like fragmentation patterns, for the chemists, typically I have to show this to prove to them that it indeed what we say it is. And so that's good. So we had a starting material, and we were able to do it. And what we saw in the genome was that this is a normal protease underneath. 1 dBi over here, and you see that it has a number of extra residues. It's called the insertion loop. The insertion loop is necessary for it to do the processing that it does. And when you look at the crystal structure, you get this extra 30 amino acid or so. They go over the top, they extend this helix here, and they go helix, turn helix, and then back. So that protects the active site from water. So it's a really nice enzyme. It's a very simple enzyme, very primitive, and again, very flexible. So what does it do? It does three things. First of all, it recognizes this aspartic acid residue in the signal sequence. And it recognizes two basic residues here, lysine, and another arginine somewhere else. So it holds the enzyme. And the way I'd like to show is it holds the enzyme, the substrate like that. Okay? Then it has to be bent to fit into the active pocket. So what you can see is this here is a very bulky set of residues that sit in the way. Normally, this substrate would bind in a linear sense like that. In this case, it has to go in the bent conformation, so it has to have a residue here, either a proline or a thiazolene, that allows it to bend in the middle. So only the bent substrate comes. It comes in bent, sits in, gets grabbed, gets cleaved just here, and then it goes around on itself to make the final product. So it's a very primitive but cool enzyme. And the last thing that it does is prevents water from getting in. So this big plug here, which is the AYD sequence, prevents water from coming in and causing hydrolysis, so you don't get the wrong product. You get only a cyclic product. So really, it's a very simple enzyme. It causes proteolysis. There's an equilibrium here. It waits for the C-terminus. You can't see it. Sorry, bad picture. You wait for the C-terminus to come around, hit the the N-terminus to come around to hit the C-terminus, and make a product. And on this side, if water gets in, and we can prove that. We can take away the lid, we can take away the recognition, and it makes a linear peptide, a fragmented peptide. So it's a very simple process. So the other thing we discovered beforehand is that the heterocycles here are necessary for the thing to bind, to loop around in a circle. So the processing has to happen before this, because otherwise it doesn't efficiently bend. So you get a low yield. With this, and this one we predict that it will go around in a circle to make a macrocycle very quickly. So also to show that you can use non-natural residues in this. So if you put in this very unusual looking thing here, it still makes a cyclic peptide. So you can put in things that are totally not amino acids to make a cyclic product. And I'll show you more of that in a minute. So this is a technical question. We still don't know. I have an argument with my colleague Jim Naismith. I think it's still spontaneous. He says it's not spontaneous. The key is that when an experiment was done by Peter Wipf in America, he showed that if that is made the wrong stereocenter, and you take a base and some heat, you can make the right product, the natural product. It's really weird. So I don't know what the answer is to that question. So what we did then is we developed a process where instead of having the very slow enzyme, we took a fast protease. We put it extra because we knew the processing can be remote from the substrate. What you do is you put a protease signal here and here, so just a lysine, and then you take a fast protease. So first of all, we did a processing with the parte, the heterocyclase enzyme, which we expressed. You have a his type to be able to purify your materials. Then you do the proteolysis, you break it open on this side, then you do the macrocyclization, you get the product. So it should be really a simple process. So that was the first process we developed. And you get, for those of you who like NMR spectra, the in vitro product that we made by biosynthesis is cleaner than a natural product, identical material. Okay? So I think this is a very nice way to show that you can use these enzymes to make the product. And the oxidation was the last step. Okay? So we need to oxidize these up to here. And we have got the last enzyme, which is the oxidase, allows us to do that last processing step. It's incredibly efficient. And just to convince you that it's possible, what you get is the product has exactly the right stereochemistry. So again, this is a way to show stereochemistry. The red trace is the natural product. The purple trace is our final product. And the blue trace is before reaction. So you can show very clearly it's exactly the same stereochemistry. So everything is now possible to make the natural product with the natural stereochemistry in 5 easy steps. Again, it's a pretty amazing project. So the other one that I showed you, the prenylase, so there's related enzymes from cyanobacteria that we published. And what you see is that this particular substrate here, the amino acid, the tryptophan, is modified with a prenyl group. So again, you can do lots of modification. You have a toolbox of enzymes that can do chemistry for you. So the idea is that you take a substrate gene here, and out of that you can do different processing. You can do the same gene, same peptide. You can process with one set of enzymes that makes all the heterocycles, or that only makes heterocycles out of the cysteines. Then you can do macrocyclization. You can do oxidation or no oxidation, different oxidations, I mean, or you can do prenylations or not. So you can make lots of different products with the same starting material. You can make 6 to 8 products if you want to. So we showed that was the case. So we made 17 different products with 6 to 9 different amino acids as ring size, with 2 to 4 heterocycles, and with 11 of the 20 natural amino acids. So again, that to us was a great achievement. And Eric Schmidt published a year later a similar system where he did everything in one pot. So again, this kind of idea is what's called chemo-enzymatic synthesis. You take the genes and you take some substrate and you add them together to make a product. And I think it's a very nice process. It's not, we can make milligram quantities, so 5 milligrams or so is possible. So again, it's more than we got from the cloning. So the interesting part is that the molecules that we made fit right into this new idea. So people are looking at macrocycles as new drug molecules, but nobody could make them. So they had to have a particular set of properties. So there had to be certain molecular weight, a certain polarity, certain size, a surface area size, certain number of bonds, etc. And our molecules have exactly the right properties to fit into that new space. So we decided to start a company based on this. I'm not going to tell you about the company. The company failed last September. So we had 2 years of great time and then nothing. So we started again. So later this year, look out for a company called Gyrox, see if it works. Okay, second time. Lucky. But then we made a nice discovery. This is following Wilfred van der Donk's work. He works on ribosomal peptides. And what we were the first to publish was the structure of the heterocyclase enzyme with the leader sequence in situ. So this here and this here shows you how the leader sequence is bound. So this is the recognition domain, this is the ATP binding domain, and this is the peptide clamp. So the recognition happens here, but the chemistry happens up here, so it's far away. So if you can activate this enzyme with the leader sequence, then what you can do is you have an enzyme as a chemical reagent. So what we did is we took a linker sequence, AG, 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 10 amino acids, linked this to the main enzyme, and now the enzyme is more soluble, more stable, easier to express, and it behaves like a chemical reagent. So you just have to add the enzyme to your substrate, and it works. So now we can do new things. And this is the nice thing. This is what the company was based on. The company was based on the idea that you take a synthetic peptide, here you go, this one here, with the small sequence, AYD. You take your engineered heterocyclase, it makes the heterocycles that you want. Then you take your engineered macrocyclase and oxidase, and you get a product. This is very, very simple. So now, instead of having to use a big leader peptide, this is leader. The 35 amino acids that were over here before are gone. You only have 11 amino acids left now, and the product is 8 amino acids. So it's pretty nice as a process. So the first showed that we could do this. We had a non-natural amino acid in here. We have a heterocycle. So you basically take your enzyme, you make the heterocycle, you add your macrocyclase, and you get your product in the same reaction vessel. So we changed the pH from 9 to 8.1. That's all you have to do. So you shift the pH, you add the new enzyme, and you get a product. So again, just to show you can do that, the reason you want that there is to do more chemistry. So now you can do chemistry, biochemistry, followed by chemistry. So you have the best of both worlds. So this is because I started off being a chemist, a synthetic chemist. So we made a library of compounds against P-glycoproteins. This is the first enzyme we wanted to aim at. Okay, so this is nice. It's a very similar library because it is based on this molecule, which is the active molecule. So we decided, so you can see the Riptide was our company name, so Riptide Molecules. And what we found was that the natural product is still the best. So here you have our standards, patelamide D. Here you have our compounds, and we never get better in nature, which is a pity. But it shows us that we can make really bad molecules too, ones that are completely inactive. But the point is that you can get what's called structure-activity relationship. We could show that we could make a family of molecules that had activity that are different from the starting material. When we showed this to industry, they said, can you make 800? And I went, ooh, tricky. So 800 molecules is hard to do. You need a lot of robots and things to make it. So we didn't have enough money to buy the robots, so we stopped the company. But now we're trying again with new funding. But other things interesting. So again, non-natural amino acids here. You get the nice, rather wet NMR spectrum. But this here is interesting because that can react. So this is an interesting study. So you take your substrate cyclic peptide that you've generated, and you can add a fluorescent linker. Right? So then you can see where it is in the cell. So these are special permeabilized cells. So it goes through the cell membrane, into the cell, and you can do now fluorescence microscopy to see it inside the cell. So it's very good for any study where you want a peptide that's cyclic with something that's reactive to get a product that is fluorescent that you can see inside a cell. So it's a general process, but I think the idea is good. So we started to play around with the system because it's so flexible. What you can see is that you can add non-natural linkers, you can add sugars, you can add long fatty chains, you can add these polar chains. And again, you can see really nice products coming out of this. And this is our selling point. We can make products that only have 3 amino acids in them, and have lots of other things in there. Because the enzymes are so flexible, they can do the reaction here. The heterocyclase still works, the macrocyclase still works. So again, all of this is very cool process. So the whole end product that we want to do is we want to show the best of chemistry. You make the synthetic substrate. You have some enzymes. This is a toolbox of enzymes that we have right now that can do heterocyclization of just cysteines, heterocyclization of cysteines, serines, and threonines. The macrocyclases, we have 9 different macrocyclases now that work all the way from 6 amino acids to 30 amino acids. So we have the whole range. We have different oxidases that do different things. We have prenolases that do different things, so you can then make your initial products. And then we can show already that you can do chemical modifications to make further products. So again, for medicinal chemistry, what you want is you want to be able to change something late. So it's called late modification. So we have a chemical handle like this. You can add another group to it. You can change the polarity. You can change the chemistry later on. So it's a very nice process, I think. So the final step is then, can we do this again in the cell? So we've deconstructed the system into small pieces. And now can we take those engineered genes and put them back into a host? So we have genes now from cyanobacteria of different types, and some bacteria, plants even now. So the point is, can we reconstruct a system inside a host that contains DNA from different organisms that works together? The answer is we're working with a company called Ingenza and now University of Oxford. Jim Naismith has just moved to there. But Ingenza is a cloning company. So they're making us constructs that contain, on 2 duet vectors, containing the substrate gene and some of the processing genes. And the eventual aim is to have the substrate gene on a separate plasmid. So you can introduce, so you have a cell that makes the factory, but then the substrate gene is put in later. So what you can do is you can have any processing you want. So the processing gene may be saying it makes 2 heterocycles, it makes a prenylation, it makes a macrocycle, and you put your substrate gene in, which is just 8 amino acids, and it makes that product for you. So the idea is that. So what we have at the moment, after 3 years of, or 2.5 years of 6 people working on this, we have a system that is completely working in the fact that all the genes are there, all the genes are expressing. We can see that. We can see some of the substrates being processed, but we can't see the final product. So the problem is that this product here is not visible yet. And the reason probably is that the cells don't live for long enough. The E. coli lives for maybe 24, 48 hours before it dies, and this last step takes 4 days. So what we've done is we've taken a plant enzyme, which is much faster for the last step, which only takes 1 hour or so, to put inside the cell, and now we're testing that. So the last step is really the critical one. If that works, then we have a system that can be producing this. So at the moment, I would say if we get production, that's already very good. If we don't get the high production, at least we've shown we can do it, and we can then engineer the system to produce the high amounts of material at the right time. So just to finish up, we can make macrocycles of different sizes with heterocycles at different states. We can also do prenylation and methylation. We can make things that no one else can make, which is, again, something you're trying to do for drug discovery. We can use synthetic substrates. We can use unnatural substrates, unnatural amino acids. Again, that's very cool. We can do further tailoring. So you can see with the fluorescent probe added, you can add chemistry at the end there. You can make analogs. If you have a substrate, so we showed already with the P-glycoprotein, you can make analogs of a known molecule to test your theory. So at the moment, we can make milligram quantities of enzyme, but we can produce the enzymes in gram quantities. So we went to a production company, and we can now produce 7 grams of one of the enzymes per liter of cells. So it's incredible overproduction. And we're generating this production platform. And the idea behind the hybrid pathway is that you could generate up to 30 amino acid peptides with modifications as sort of small antibodies. So this is the work of a lot of people, and I have to really thank a number of people. The first one is Maggie Smith. She had faith in me when nothing was working. So for 6 years, we had no results published at all in this area of research. And she had faith that it would work. And the postdoc who did all this work was Wael. Wael is a fantastic scientist. He is the driving force behind it. If he says something doesn't work, it doesn't work. He's tried all possible conditions. But then again, he had a very good working relationship with Jesko, who was the crystallographer, the first crystallographer to work on this. He had a DFG grant. And beyond that, all these postdocs, all these PhD students, the collaborators at St Andrews, again, all the postdocs there, all the PhD students there, and our collaborators in Finland have been instrumental in making all of this work. So it's not the work of a single person. I like to present it as a group work. And of course, the funding came from a lot of different people. So I have to really thank two agencies. The most important is the Labium Trust. It is a UK funding agency that funds high-risk research. So if everything is going badly and you can't get money, you write to these people and you say to them, the Research Council won't fund it, I can't get money from industry. This is why it's high risk, but it would pay off if it worked. And on 2 occasions in Australia, they funded my research for 2 years for research afterwards, and in 2010, they funded the research that went on to the crystal structure. So again, when it was critical, they funded it. Then we got money from the BBSRC, from Merck Sharp and Dome through the SALSA, Deutsche Forschungsgemeinschaft, Scottish Enterprise, Framework 7, a little bit of work on the analysis done there. But the most important one is Jim Naismith and I got an ERC advanced grant together on this. So again, this is a 5-year project with 3 postdocs, 3 PhD students. And it's been tremendous because it's allowed us to develop a lot of the basic science that goes behind the applications. So thank you very much. Hope you enjoyed that.
Thank you very much. Very interesting results. And I don't know if there are some questions from the audience.
I think I'll start. Thank you, Marcel. I was wondering, can you talk a bit more about the bioactivity of these molecules that you're producing? I mean, you know, like anything, what are you going for?
So the first thing is that initially the idea was against P-glycoprotein, and that is the drug efflux pump. So it's like a funnel-shaped thing inside the cell that turns over, takes the molecule and turns back again and ejects toxins. And it was believed at the time that inhibiting that protein could lead to reversal of drug-resistant cells in vivo. Since then, there's been a lot of problems in the clinical trials of that approach. But secondly, the real key behind this is that the change in drug discovery is happening. So the original mechanism of lock and key, where you have a small molecule that fits in an active site that is able to be inhibited by biological processes, only a very small number of enzymes that are doing that. So this is maybe 500 different enzymes in the body have that simple rule. But about 30,000 other processes in the body are based on protein-protein interactions. So the big proteins come together like this, and we need big molecules to inhibit that. So what you need is these big cyclic peptides, and nobody can make them, so nobody can design them either. So we have an approach that allows us to build these molecules, and now we're designing them too. So I have a very talented student who's doing modeling right now, and he's building models of these cyclic peptides that would interfere with the process, and he's building the molecules and testing them. So he has 4 different approaches. Most of them are immune system based. So TNF-alpha, which is for arthritis. He has multiple sclerosis proteins and other ones. He's modeled them, designed the molecules, built the molecules, and now testing. And out of those, he will choose one to develop further, see if it's active. So if it's active, I hope that one of them is at least active out of the 4. He will develop the next stage. And that's, again, it's the change in drug discovery. So our big problem with our company was that when we were working on this, there was a Japanese company called Peptidream, and every time we went to a big company to speak to them about it, they said, we just signed a deal with Peptidream. So every time we were too late, and what we were offering was not enough. They were offering 1 billion molecules, we were offering 100 molecules. So again, the scale was very different. But now the realization is that actually what you need is not 1 billion molecules you can't see, but you need 100 molecules you can see enough of. So again, we're working on that again. So overcoming the technical challenge of making 100 molecules efficiently and then testing against these different diseases. And again, it's exciting space to be in because the companies have come back to us now and say, can you still do that? Because we have, you know, they have a hit, but they can't produce the materials. So now we can produce the materials for them using our process. So it's, yeah, So that's the reason for doing it.
No, but I was wondering about the bioactivity. Have you, like...
Yeah, so I mean...
any specific type of forage, because at the beginning, like...
Yeah, so initially they're all cytotoxic, so they're really cytotoxic. So they've tested them in animals, and they have no specificity, no selectivity, they just kill everything. So the animal trials failed, basically because they killed the animal. So yeah, so the NCI, so we have a paper that summarizes all the bioactivity. But they had a number of different types of activity, I think DNA polymerization inhibition, P-glycoprotein inhibition, but all of them pretty much just kill the cell. And some of them are really toxic. So one of the potential ideas was to use them as antibody drug conjugates, ADC, because the toxicity was down in the low nanomolar and subnanomolar. But perhaps not. I think the idea of making cyclic peptides for drug protein-protein interactions is probably the way to go for us. Because again, we can modify things now, change things. So we're not going to rely on the basic activity of the molecules, but to just go for completely new activities and design the molecules. So I'm a synthetic chemist by training, so I came into natural products and then genetic and then molecular biology and now back again to synthesis.
Thank you again.
Okay.